Hey guys, today we're going to go over Unit 1.1 of the IB syllabus, which is the Introduction to Cells. First, we're going to go over the cell theory, which states that all living things are composed of cells. Cells are the smallest unit of life, and all cells come from pre-existing cells. Now for the first point, how did scientists discover this? Well, what they did is they took various organisms, such as onions, animals, and they put them under a microscope and they saw organelles such as the cell plasma membrane, the nucleus, and cytoplasm, and this was the evidence that all living things were composed of cells because the scientists were able to identify these organelles in every single organism they looked at. Second point is that the cells are the smallest unit of life. How do scientists know this? Well, they just say no living organism has been found that's not made out of cells, so cells must be the smallest unit of life. What about viruses? First of all, they're not living, they don't have a metabolism, and they cannot reproduce without a host cell, so even though they are smaller than cells, they're not living. What about organelles? They can't live outside the cell, so organelles are smaller than cells because they are inside them, but since they cannot live and sustain life outside the cell, we do not classify them as living organisms, which does not contradict the statement that cells are the smallest unit of life. Alright, the last point is that all cells come from pre-existing cells. Scientists know this through Pasteur's test of spontaneous generation. I'm not going to go over this in this video, but I'll go over it in Unit 1.5. So look out for that if you guys are interested. Alright. So the exceptions to the cell theory. Before we go over the exceptions, we're going to go over two words, trend and discrepancy. You guys probably know what it means already, but a trend is a prevailing tendency or generalization, while a discrepancy is the exception to a general trend. The first exception we're going to look at is striated muscle cells. They're the muscle cells that are stuck to your skeletal bone, so they're also called skeletal muscle cells. They're multinucleated, which means that they have multiple nucleuses, and they're very long. They're 30 millimeters compared to the norm of 0.3 millimeters. As you see, there we go. So that is one cell, but as you can see, they have multiple nucleuses and they're very long. This is an exception because most cells aren't that long and they only have one nucleus and there's only one nucleus to each cell. Alright. Another exception is the aseptate fungal hyphae. This is unique in that no walls separate the regions of the hyphae, which means that no walls separate each cell of the hyphae. You can see that here. So septate hyphae have these walls that separate each cell. So you can see there's one nucleus per each cell, and there's a septum that separates each and every cell. But in aseptate hyphae, you can see that there aren't these walls. There aren't these septums that separate each cell or nucleus from each other, and that proves to be an exception of the cell theory. Right? The last exception is giant algae that looks like that. It seems to be one big cell. It's up to 100 millimeters in length, and as I said, most cells are only 0.3 millimeters. That's, almost 1,000 times larger. That's a great difference. Those three were the three exceptions to cell theory. You guys will probably need to know that for a test or something, so make sure you write it down. Alright. Now we're going to go over two organisms. single-celled organisms, the Paramecium and the Chlamydomonas. So, what's the Paramecium? It's a heterotrophic unicellular organism. Heterotrophic means it doesn't produce its own food, and unicellular means, well, it's only composed of one cell. So, single-celled organism. It looks like this. You guys probably would want to know how to draw this, so if you guys want, pause the video here to take a look at the diagram on the left. And on the right is just a micrograph for the Paramecium. Alright, and the Chlamydomonas is an autotrophic unicellular organism, which means it produces its own food, single-celled. Looks like that. On the right is the diagram. You guys would probably want to know this too, so pause the video here if you guys want to copy the diagram down. And on the left is also just a micrograph. Alright, so now we're going to go over the functions of life. Remember, Mr. H. Gren. The M stands for metabolism, R stands for response, H stands for homeostasis, G stands for growth, R stands for reproduction, E stands for excretion, and N stands for nutrition. We're gonna go over each and every one of these, each and every one of these functions of life and compare these functions that occur in Paramecium and Chlamydomonas. So, what is metabolism? Metabolism is the chemical reactions inside a cell. There we go. In Paramecium metabolism occurs as they produce enzymes that catalyze many different chemical reactions in the cytoplasm. Likewise, Chlamydomonas produce enzymes that catalyze many different chemical reactions in the cytoplasm. It's safe to say that metabolism occurs exactly the same in both organisms. Not exactly the same, but the general idea is similar. Alright, so response. What's response? It's reacting to stimuli, and as in stimuli mean, light or an obstacle, or something like that. In Paramecium, when Paramecium contacts a solid object, the cilia, as shown in the diagram, be in a different direction to reverse the direction. So let's say the Paramecium bumps into, a rock or something, then the cilia, the cilia right here would propel the Paramecium in the opposite direction so it could move away from the rock. And for the Chlamydomonas, the eye spot senses the brightest light and swims towards it. The eye spot's right there. And the flagellum is responsible for movement in Chlamydomonas. Alright. So homeostasis. Homeostasis is keeping conditions inside an organism within tolerable limits. These conditions include temperature or pH, for example. Right, so in Paramecium, water content is maintained because contractile vacuoles, right there, remove the excess water. Likewise, it's the same thing, Chlamydomonas, the contractile vacuoles are here as well. Alright, so growth. Growth, pretty straightforward, irreversible increase in size. Paramecium, size and dry mass increases due to absorption of organic matter in food. They don't produce their own food, so they must absorb organic matter in the food in order to grow. On the other hand, Chlamydomonas, they produce their own food through photosynthesis, and they absorb minerals as well, and that leads to size and dry mass increase. Right? Reproduction is producing offspring. It occurs sexually and asexually. in Paramecium and Chlamydomonas, in most single-celled organisms, they usually are asexual. So it's the same for both. It could occur asexually or sexually. Asexually, it would be called mitosis or binary fission, and sexually, it would be meiosis. We'll learn about mitosis and meiosis in future videos. So if you guys want that, make sure to subscribe to my channel. Excretion. Excretion is the ridding of waste. So CO2, carbon dioxide from cellular respiration, diffuses out of the cell in Paramecium, whereas oxygen from photosynthesis diffuses out of cell. And if you guys took a normal biology, I don't know, in middle school, you guys probably know that most cells go through cellular respiration, even though it does photosynthesis. So when Chlamydomonas is doing cellular respiration, it will diffuse CO2 as well. But those are the main points. Nutrition is obtaining food for growth or energy. So the Paramecium, it ingests and digests smaller organism food vacuoles. This process is called endocytosis. Chlamydomonas, as I said earlier, produces its own food by photosynthesis using chloroplasts. Right. So the limits to cell size. Why are cells so small? Well, a larger surface area means a faster exchange of materials, and a larger volume means more metabolic reactions. Metabolic reactions require raw materials and produce waste, so larger cells need a larger surface area because faster exchange of materials needs to happen. When volume increases, surface area does increase, but the surface area to volume ratio actually decreases. As you can see, if you guys do the math, pause the video right here to figure it out for yourself. The two cubes are obviously different. The cube on the left has side length 2, while the cube on the right has side length 1. The surface area to volume ratio of the cube on the left is 3, while the surface area to volume ratio on the cube on the right is 6. And as the cube increases, you guys will find that the surface area to volume ratio continues to decrease. Therefore, as a cell grows, its ability to gain or lose materials or waste does not increase enough in comparison to the rate of metabolic reactions. Yes, the rate of exchange of materials does increase as the volume of the cell increases. However, it does not increase as fast enough as the volume and the amount of raw materials and waste produced and needed. So, therefore, the cell must either stop growing or divide. Some cells develop specialized structures to increase surface area, such as elongated muscle cell, as we saw earlier, or the nerve cell axon. So, emergent properties. What are emergent properties? They arise from the interaction of the component parts of a complicated structure. Made it sound a little complicated, but as you guys see examples, you guys will figure it out. Anyways, multicellular organisms have properties that emerge from the interaction of cellular components. Interactions between organelles result in life. Interaction between nerve cells result in consciousness. Interactions between water molecules result in surface tension. These are all examples of emergent properties, and you guys will need to know that for your test. So cell differentiation. Cells are differentiated by changing which genes are expressed. All cells of the body have the same DNA, but as you see, your skin cells and your heart cells are bound to look different. And how do they do that? By changing which genes are expressed. So this is essential in embryonic and new tissue development because, as I said, all the cells in the body have the same DNA, but all cells need to differentiate into their different functions, and yeah. So stem cells are cells that have retained their capacity to differentiate along different pathways. Types of stem cells include totipotent, which can give rise to an entire organism, so it would be an embryo. A pluripotent cell, they can give rise to any type of cell. And induced pluripotent cells are differentiated cells that have been reprogrammed so that they can give rise to any type of cells. Adult stem cells are induced pluripotent cells. So, stem cells for disease treatment. Okay. So, the source of embryonic stem cells, wait a minute, let me back up. Okay, so 3 types of cells can be used for disease treatment: embryonic stem cells, cord blood stem cells, and adult stem cells. The source of embryonic stem cells is embryos, so they're created by destroying specially created embryos in a petri dish to get stem cells. And from the umbilical cord of a newborn, cord blood stem cells can be obtained. They're easily acquired, but only a limited quantity of cells can be produced. Adult stem cells are very difficult to obtain since they are few and most are found deep inside of your tissues. Alright, the potential or the capacity to divide. For embryonic stem cells, this is unlimited. Cord blood, limited, and adult stem cells, limited. The potential to differentiate, which means potential to turn into any cell of any tissue in the body. The embryonic stem cells are pluripotent, which means that it can turn into any cell in the body. Whereas cord blood stem cells and adult stem cells are limited to a few cell types. Alright, so the compatibility with recipient. Embryonic stem cells come from other individuals, so they're likely to be incompatible. Cord blood stem cells, if given back to the same individual, so let's say when I was born, the doctors took my stem cells from my umbilical cord, and then now I need, a bone marrow transplant. Then those stem cells can be used and it would be fully compatible. And adult stem cells are fully compatible because they, like cord blood stem cells, go to the same individual. The risks of embryonic stem cells: rejection is possible, as I said earlier, and they have a high chance of developing into tumor cells, which we all know cause cancer. Cord blood stem cells also have risks in that pre-leukemic cells may be present in cord blood. Adult stem cells have less growth potential than embryonic stem cells, so that's not really risk, but it's just one of the not benefits, I guess. Benefits. Embryonic stem cells can be used for many different tissues. Cord blood stem cells have no rejection, adult stem cells have no rejection, and there's less chance of tumors developing. That would be the same for cord blood stem cells. I don't get why I didn't put that there, but yeah. Less chance of malignant tumors, blah blah, that is the same. That also, I cannot think of the word, that also retains to the cord blood stem cells as well. Alright, so ethical issues. Embryonic stem cells are very problematic because some people argue that creating embryos and then destroying them for stem cells are unethical. Both cord blood stem cells and adult stem cells don't really have ethical issues, especially since for cord blood stem cells, they would be discarded anyway. Okay, ethics of embryonic stem cells. People that support the statement that embryonic stem cells are unethical would state that an embryo is a human life. And yes, we acknowledge that embryonic stem cells have immense benefits. However, these benefits do not justify taking a life. The people that support the usage of embryonic stem cells would say embryos are just balls of cells. They don't have a nervous system, therefore they don't have consciousness. They don't really know they're being killed. Also, a large number of embryos are produced by test tube fertilization, artificial insemination, and they're never implanted into the mother's body. Therefore, they don't get the chance of life. They're going to be killed anyways. So, it's better to use them for stem cells rather than just killing them. Alright. Stargards disease is an example of a disease that can be treated using stem cells. So, what is it? It's a genetic disease causing macular degeneration in the eye, causing damage to photoreceptor cells in the retina, leading to blindness. The procedure of treating Stargards disease using stem cells is as follows. So embryonic stem cells are made in the lab to differentiate into retina cells. These retina cells, thousands of them, are injected into the patient's eyes. The patients show improved vision and they report that there are no harmful effects. However, more trials need to be taken to optimize long-term safety. Another disease that can be treated using stem cells is leukemia. Leukemia is a type of cancer in which an abnormal large number of white blood cells are produced due to mutations in genes controlling cell division. The procedure is as follows. So, bone marrow is extracted from a bone of a donor. Healthy stem cells are extracted from this bone marrow and stored somewhere else. Chemotherapy kills cancerous cells in the patient. All the stem cells in the bone marrow of the patient are destroyed in the process. Now, since there are no stem cells in the bone marrow, healthy stem cells are injected into the patient's blood. These healthy stem cells migrate to the bone marrow, and there they produce healthy white blood cells. The risks of leukemia the risks of treating leukemia with stem cells include the rejection of cells from donor and the presence of cancerous cells in the donated healthy stem cells. So the last thing we're going to go over is microscopes and magnification. The invention of electron microscopes led to greater understanding of cell structure. For example, it let scientists identify various organelles, smaller organelles that they weren't able to see with just a light microscope. And I guess the IB thought it's important for us as students to know how to manipulate data and look at micrographs and see how much it's magnified and such. So therefore, here's the equation for magnification. Size of image over size of specimen equals the size of scale bar on an image over the number on the scale bar, which equals magnification. You guys will probably be required to find the magnification. I included two problems. So let's say the size of the image, so I measured the image with the ruler. It turned out to be 15 millimeters. And the size of the specimen, the actual size of this cell, is, let's say, 15 nanometers. If you do the math, plug it in the equation, you get that the magnification is 300 times. I probably should have said that you guys should pause the video and do it yourself, but this is pretty straightforward. If you guys need more practice, just do more problems on magnification. They're all online. Another example, let's say the size of the scale bar in the image is 20 millimeters and the number says it's 25 nanometers. Do the math, you get 800. Easy. Alright, thanks for watching. Make sure to subscribe for more videos on biology, physics, or math. Thank you.